Hello, 7.3 lecture coming up. It's about cell transport. And I don't mean transport of organelles, I mean molecules. So we'll be learning about that. It's osmosis. So this is not a Gary Larson fireside. It's the look on the bright side. With only one cell, how much can go wrong? It's cute. All right, our goal is to relate the structure to function for the components of plant and animal cells. Also explain the role of cell membranes as a highly selective barrier and active and passive transport. Okie dokie. Our objectives are to be able to describe passive transport and describe active transport. That sounds easy, right? Oh, hang on to your brains and stick with me. All right, let's think about it. When thinking about how cells move materials in and out, it can be helpful to think of a cell as a nation. The boundaries of the nations are its borders, and nearly every country tries to regulate and control the goods that move across those borders. Cells have their own borders, which separate the cell from its surroundings and also determine what comes in and what goes out. So how can a cell separate itself from its environment and still allow material to enter and leave? Let's find out. Let's talk about passive transport first. All right, so if every living cell exist in a liquid environment. One of the most important functions of the cell membranes is to keep the cell's internal conditions relatively constant, homeostasis. And it does this by regulating the movement of the molecules from one side of the membrane to the other. Remember, everything is molecular motion just because it has heat. So high and low concentrations help things to move. Diffusion. So within the cytoplasm of a cell is a solution of many different substances dissolved in water. In a solution, solute particles tend to move from an area where they are more concentrated to an area where they are less concentrated, and more concentrated to less concentrated. The process by which particles move from a high area concentration to an area of low concentration is known as diffusion. And diffusion is the driving force behind the movement of many substances across the cell membrane. You can think about diffusion as very simply as if I lit a match in the classroom, I would smell it first, but eventually the whole classroom would smell of the match. All right, let's look at a step-by-step illustration of diffusion. Suppose a substance is present in unequal concentrations on either side of the cell membrane. If a substance can cross the cell membrane, its particles will tend to move toward the area where it's less concentrated until eventually it is equally and evenly distributed. At that point, there you go, not that one. At that point, the concentration of the substance on both sides of the cell membrane is the same, and it's called an equilibrium is reached. Even when an equilibrium is reached, though, keep in mind the particles of solution, because of heat motion, will continue to move backwards and across the membrane in both directions. Because almost all equal numbers of particles move in each direction, there's no net real change in the concentration on either side. Diffusion depends upon the random particle movements. Substances diffuse across membranes without requiring the cell to use additional energy. The movement of materials across the cell membrane without using cellular energy is called passive transport. Some molecules can go through the cell membrane, but most have to go through the membrane through carriers. And that would be facilitated diffusion. Cell membranes have proteins that act as carriers or channels that make it easy for certain molecules to cross. Molecules that cannot directly diffuse across the membrane pass through special protein channels in a process known as facilitated diffusion. Hundreds of different proteins have been found that allow particular substances to cross cell membranes. The movement of molecules by facilitated diffusion does not require any additional use of the cell's energy. So here is not a recap, but an introduction to what osmosis is to help you wrap your brain around it. So let me go back to this one actually first. Inside the cell's lipid bilayer is the hydrophobic or water-hating part, the lipids there. And because of this, the water molecules have a really tough time passing through the cell membrane. Many cells contain water channel proteins called aquaporins. Pore, because, they go through those pores, and they're water. They're for water. That allow the water to pass right through them. And without aquaporins, water would diffuse in and out of the cells way too slowly for the cell to function properly. So the movement of water through cell membranes by facilitated diffusion is an extremely important biological process. It's the process called osmosis. And if you ever saw that movie Osmosis Jones, it's several years old now. It has nothing to do with this. So osmosis is a diffusion of water through a selectively permeable membrane. Osmosis involves the movement of water molecules from a higher area of high concentration to the area of low concentration. A few vocabulary words for you. Tonicity. Tonicity is comparing the solute concentration of two or more solutions. So when you're doing tonicity, you're comparing them. And there's three different states of tonicity. You have isotonic, when they're equal on both sides. You have hypotonic, when there's more water on one side and less solute. And there's hypertonic, when there's less water on one side and more solute. And osmosis, again, is the movement of water molecules from hypotonic to hypertonic. So in an experiment we see here, the barrier is permeable. This barrier right here is permeable to water, but not to sugar, because the sugar molecules are larger, but the water molecules can move through it. This means that the water molecules can pass the barrier, but the sugar cannot. There are more sugar molecules on this side of the permeable barrier than on the left side. The right side has more. Therefore, the concentration of water is lower on the right, where more of the solution of sugar has sugar in it. So what's going to happen is there's going to be a net movement of water into the compartment containing the concentrated sugar solution. Water will tend to move across the barrier until an equilibrium is reached. These little the water, not the sugar. The water will move across the barrier until the equilibrium reached. So it actually pushes up against gravity in effect too. At that point, the concentration of water and sugar will be equal. So when the concentration of water is equal on both sides of the membrane, the two solutions will be what's called isotonic solution, because they're same strength. The more concentrated sugar solution at the start of the experiment was hypertonic, or above strength, compared to the dilute sugar solution. The dilute sugar solution was hypotonic, or below strength. So for organisms to survive, I gotta go back to this one, they must have a way to balance the intake and loss of water. The net movement of water out of or into a cell exerts a force known as osmotic pressure, turgor pressure. Because the cell is filled with salts, sugars, proteins, and other molecules, it is almost always hypertonic to fresh water. Here's osmotic pressure for turgid. So as a result, water tends to move quickly into a cell surrounded by fresh water, causing it to swell. Eventually, the cell could actually burst. All right, in a hypotonic situation, in plants, the movement of water into the cell causes the central vacuole to swell, pushing the cell contents out against the cell wall. It gets nice and fat. And since most cells in large organisms do not come in contact with fresh water, they are not in danger of bursting. Instead, the cells are bathed in fluids such as blood that are isotonic and have concentrations of dissolved materials roughly equal to those in the cell. Cells placed in an isotonic solution neither gain nor lose water. This goes back and forth equally, isotonic. In a hypertonic solution, water rushes out of the cell, causing animal cells to shrink and the plant cell vacuoles to collapse. And that's when you see the plants go limp. And it's called flaccid cell. In the animal cells, when they kind of shrink up, and it's called to crenate. All right, it gets shrunk up. Some cells, such as eggs laid by fish and frogs, must come in contact with fresh water, obviously. And these types of cells tend to lack these water channels, these aquaporins. So as a result, water moves into them so slowly that the osmotic pressure does not become a problem. Other cells, including those of plants and bacteria that come into contact with fresh water, are surrounded by the tough cell walls that prevent the cells from expanding even under like tremendous osmotic pressure. Notice how the plant cell right here holds its shape in hypertonic solution, while the animal red blood cell does not. It gets fat and bloated. However, the increased osmotic pressure makes cell shells extremely vulnerable to injuries in their cell walls. So the plant is what you're going to call a turgid cell, and the animal cell has become lysed, L-Y-S-E-D. There you go. Okay. 2 more slides about osmosis here. The first is showing plants in the turgid state, and then a normal state. Well, not quite normal, it's losing its water. And then eventually you can see what happened. The vacuoles here are very swollen. The plant's nice and happy and tall, and it becomes to wilt as the water leaves the cell. Another one shows red blood cells. The first is the red blood cell is a hypotonic solution, and the water rushed into it by osmosis, causing it to become what's called a lysed state. Right here, they're lysed fat. They don't function very well like that. In the middle one here, it's in a hypertonic solution. All right, so the water had rushed out, and the cells have now become what's called an acrinate state. On the end, these are perfect little red blood cells, and they are in their isotonic state. We are going to be doing this lab in class. We're taking water and sucrose, and you're going to see an experiment with osmosis some more. So I'll help you understand it better. So describe passive transport. Passive transport is the movement of materials across the cell membrane without using any energy, cellular energy. It's passive transport. Okay, let's talk about active transport, a little more simpler. Active transport. All right, sometimes must move materials against a concentration difference. All right, the movement of materials against a concentration difference is known as active transport. Active transport requires energy. The active transport of small molecules or ions across the cell membrane is generally carried out by transport proteins or protein pumps that are found in the membrane itself. Large molecules and clumps of material can also be actively transported across the cell membrane by a process known as endocytosis and exocytosis. The transport of these larger materials sometimes involves changes in the shape of the cell membrane. So endocytosis is like this large inside. It brings it in. It's bringing things in, endo, and exo is it's fusing and taking it out. Small molecules and ions are carried across the membranes by proteins in the membrane that act like pumps. Many cells use proteins to move calcium, potassium, and sodium ions across the cell membranes. Changes in protein shape seem to play an important role in the pumping process. A considerable portion of the energy used by cells in their daily activities is devoted to providing the energy to keep this form of active transport working. The use of energy in this system enables cells to concentrate substances in a particular location, even when the forces of diffusion might tend to move these substances in the opposite direction. Larger molecules and even solid clumps of material may be transported by movements of the cell membrane known as bulk transport. I messed up here. Bulk transport can take several forms depending on the size and shape of the material moved into or out of the cell. So let me finish this. I missed something, so let's go back to endocytosis. Endocytosis is the process of taking material into the cell by means of infolding, brings it in, or pockets of the cell membrane. The pocket that results breaks loose from the outer portion of the cell membrane and forms a vesicle or vacuole within the cytoplasm. Large molecules, clumps of food, and even whole cells can be taken up by endocytosis. 2 examples of endocytosis are phagocytosis and pinocytosis. In phagocytosis, extensions of cytoplasm surround a particular particle and package it within a food vacuole. The cell then engulfs it, Amoebas use this method for taking in food. Engulfing material in this way requires a considerable amount of energy and therefore is a form of active transport. In penocytosis, cells take up liquid from the surrounding area, like these little things right here, little pieces of liquid, by forming tiny pockets along the cell membrane. The pockets fill with the liquid and then pinch off to form the vacuoles, or little vesicles, within the cell. Many cells release large amounts of material from the environment by forming tiny pockets along the cell membranes. The pockets fill with I lost my spot, liquid and pinch off to form vacuoles within the cell. All right, so that's endocytosis and exocytosis, and you can read those there and study them too. Describe active transport was our objective. The movement of materials against concentration difference is known as active transport, and active transport does require energy. And there's an interesting picture for you. I'm just really tired right now. Can't even think straight.